Live आरंभ होईल. Tanmay start.
A very good afternoon, everyone. Myself, Tanmay Ghosh, would like to extend a very warm welcome to all of you present here. Honorable Principal Sir Dr. Prasanta Bhattacharyya, Coordinator I of IQAC, Professor Tanmay Bandyopadhyay, Webinar Host and IT Support Dr. Pronovi Porel, Resource Person Dr. MUKHERJEE, and my beloved organizing team. It is a moment of great privilege and honor for me to be a member of this webinar. I would like to thank Rabindra Mahavidyalaya and specially thanks to Principal Sir for supporting and opening this forum. I would like to express my sincere gratitude to the speaker for donating their valuable time with us. Today we are all gathered here for today's international webinar on combating COVID-19 crisis and its aftermath. Insights by young researchers organized by the Department of Microbiology and IQAC Rabindra Mahavidyalaya. I find it to be my pleasure in presence of our Honorable Principal Sir Dr. Prasanta Bhattacharyya. I feel privileged to extend my warm welcome to you, sir. I wish to thank you. Without your support, it could not be possible for us. Now I am requesting you to inaugurate the program. Please, sir.
Thank you, Tanmay, for. I think everyone can hear me.
Yes, sir.
Okay. Thank you very much, Tanmay, and Joydeep. Also, I'm seeing on the screen Naeem, my other teacher in the Department of Microbiology, and of course, the IQAC coordinator, Professor Tanmay Bandyopadhyay, my relentless host, Pronovi, who has been, no amount of praise, I think, is really adequate for her, is enough for her. She has been continuously at it from the time we have started developing RMV online as a platform for teaching, learning, and later on for hosting such programs pertaining to the webinar. And the other kind of logistic support that we continue to receive from the other staff and members of our college, especially the IT committee, who had recently held several meetings among themselves. And last but not the least, my guest speaker today, AMARSHI Mukherjee, who is closer home and yet physically is staying outside the kind of native ambit of ours, because he's now in the US, as I have been told and informed by the teachers of the department, and that he has agreed to come over here to deliver his very important talk and share some present research-based insights into the kind of understanding that we are developing for coping up this sort of a crisis, which is unprecedented, so to speak, in human history. To begin, I mean, as a very layman, I just come to hear this kind of thing that the coronavirus can stay aloft for hours. And now people are suggesting, who are the experts, that they may exist in tiny droplets, infecting people as they inhale, mounting scientific evidences. They suggest something like that. So the risk at present is highest, and therefore staying indoors, while it is being given as a sort of a mandatory guideline for all of us, is also becoming sort of risky kind of an affair. So that is also another reason why we are holding such kind of a webinar, and proper ventilation and the other necessary things are really the need of the hour, so that we can cope up with this kind of present danger of coronavirus. it's still very unclear how often the virus is spread via these tiny droplets or aerosols, which is a new kind of an addition to our knowledge, compared with larger droplets expelled when a sick person coughs or sneezes, or transmitted through contact with contaminated surfaces. Now, for a virus to be airborne means that it can be carried through the air in a viable form, but measles is another form of airborne virus. We all know that. And it can survive in the air for up to 2 hours. For how long the coronavirus may uh, stay alive in the air is not a contentious issue, but therefore it is only for this reason that we are having the expert with us today, and he definitely is supposed to share the present insight and the present research that has been made in the US, in the well-equipped laboratories, so much so that we can gain some ideas and also benefit from the kind of researches that are being daily carried on over there, with more sort of support, more sort of funding into it. And of course, another big problem is this aspect of physical distancing, how much it is needed, whether it is needed at all, etc., etc. The questions are being raised continually. The closer we are to an infected person, the more aerosols and droplets one may become exposed to. Now, washing our hands daily and often is still a good idea. And then what's new is that those things may not be enough. That is the thing. We should be placing as much emphasis on masks and ventilation as we do with hand washing. So this has become important, I think. And despite the many photos of people at the beaches, I rather very commonsensically try to feel that even somehow or somewhat crowded beach, especially if the day is particularly breezy, is safer than a pub or an indoor restaurant. So even outdoors, we better should wear a mask. And if we are likely to stay close to the others, and that also for an extended period of time, these are the safety measures we are bound to take, I think, or resort to. Now, open windows and doors whenever it is possible. This has been suggested by our very honorable Chief Minister in a recent video kind of video chat with some journalist. And what we may also try to do is to upgrade the quality of filters that the upscale people in our neighborhood may have for their air conditioning system. So we may adjust it so that the air gets more kind of outside. More outside air could be pumped into the system rather than the kind of indoor air getting recirculated continuously. Public buildings and businesses may want to invest in air purifiers and ultraviolet lights that can kill the virus. But despite their reputation, elevators may not be a big risk compared with public bathrooms or offices with stagnant air, where one is sometimes forced to stay or spend some long hours. If none of these things are possible, I think we should try to minimize the time that we are likely to spend in an indoor space, especially without a mask. The longer we spend inside, the greater the dose of virus we tend to inhale. So with these sort of rudimentary ideas, I, as a layman, along with so many like me, fighting this crisis, coping up with this crisis, and coming up with this new danger of coronavirus, is presently equipped with. I look up to the experts like the present guest speaker of today for providing us with more valuable insights so that our living becomes clear, and we tend to get rid of the sort of sense of crisis that we are having to deal with almost on a daily basis. So over to the coordinator Tanmay once again to take us to the next step of the present proceeding. Thank you.
Thanks, Tom sir, for your valuable information. Now it is a great pleasure that Honorable Coordinator of IQAC, Professor Tanmay Bandyopadhyay, is present among us. I would like to request to Professor Bandyopadhyay to share with valuable thought with us. Please, sir. Hello, sir.
তন্ময় ঘোষ, আমাদের মাইক্রোবায়োলজি ডিপার্টমেন্টের এক্স ফ্যাকাল্টি মেম্বার, মাননীয় অধ্যক্ষ মহাশয়, ওয়েবিনারে উপস্থিত শ্রদ্ধেয় বক্তাগণ, আমার কলেজের শিক্ষক শিক্ষাকর্মী বৃন্দ ও আমার স্নেহের ছাত্রছাত্রীরা, বেশ কিছুদিন আগে নঈম খানকে আমি বোধহয় একটু চেষ্টা করেছিলাম বলার যে এরকম মাইক্রোবায়োলজি ডিপার্টমেন্ট থেকে একটা ওয়েবিনার করার জন্য, এবং সেই নঈমের উদ্যোগ এবং তারপরে জয়দীপ ডিপার্টমেন্টের সবাই এটা উৎসাহিত বোধ করেন এবং ঠিকঠাক করেন যে হ্যাঁ একটা ওয়েবিনার আমরা খুব তাড়াতাড়ি করব। তো আমাদের এক্স ফ্যাকাল্টি অর্থাৎ তন্ময় ঘোষ আমাদের তো কলেজে চাকরি করতেন, কিন্তু উনি এখন চাকরি পেয়ে চলে গেছেন কলকাতার একটি সুপ্রসিদ্ধ কলেজে, এবং তার সাথে যোগাযোগ করতে তিনি যে এতটা উদ্যোগ নিয়েছিলেন এবং আমাদের কলেজকে যে এতটা ভালোবেসে উনি পুরো সময়টা দিয়ে এটার ব্যবস্থা করেছেন এবং প্রায় একটা ইন্টারন্যাশনাল লেভেলের সেমিনারে এটা করে দিলেন, চেষ্টা করলেন, তার জন্য তাকে অসংখ্য ধন্যবাদ।
থ্যাংক ইউ স্যার।
আর আমরা যে ওয়েবিনার উপস্থাপন করতে চলেছি এটা আমাদের কলেজ ওয়েবিনার সিরিজের 7ম অধিবেশন। এর সঙ্গে সঙ্গে আমি তো প্রণবী ম্যাডাম, ডক্টর প্রণবী পোরেল, যিনি এটার হোস্ট আজকের এই ওয়েবিনারের, তিনি প্রায় প্রতিটাতেই হোস্ট এবং এত সময় দিয়ে এত যত্ন করে এগুলো করে যাচ্ছেন যে আমার মনে হয় কোনো কলেজ এতটা সাকসেসফুলি রান করতে পারছে কি না এই পিরিয়ডে, লকডাউন পিরিয়ডে বা আনলক 2 পিরিয়ড পর্যন্ত। আমরা বোধহয় মে মাসের গোড়ার দিক থেকেই এই সিরিজ অফ ওয়েবিনারটা শুরু করেছিলাম এবং আমাদের প্রিন্সিপাল স্যারের সুপরামর্শে এবং ওনারও অক্লান্ত প্রচেষ্টা, এইভাবে তো বাড়িতে বসে এটা করে যাওয়া এত সহজ ব্যাপার নয়, তাই জন্য ওনাকেও আমি আবার ধন্যবাদ জানাই। বর্তমান সময়ে আমরা যে নোভেল করোনা ভাইরাস এর আক্রমণে প্রায় সকলেই দিশাহারা, সেই ভাইরাসটি সম্পর্কে আমরা এই আলোচনা সভায় আরো বিস্তারিত জানার চেষ্টা করব আমাদের সঙ্গে যারা রয়েছেন নব্য গবেষকদের কাছ থেকে। এখন প্রায় 4 মাস, বলতে গেলে 4 মাস অতিক্রান্ত হয়েই গেল ধরে এই বিশ্ব ত্রাস ভাইরাসটি নিয়ে আমাদের পথ চলতে হচ্ছে। তবে নানান মাধ্যম থেকে যেটুকু জানতে পারছি তাতে মাঝে মধ্যে খুবই ধন্দে পড়ে যাচ্ছে। একবার কোথাও বলা হচ্ছে যে আগামী এত তারিখের মধ্যে পৃথিবী থেকে এই ভাইরাসটি নির্মূল হয়ে যাবে। তারপরে খুব সাবধানবাণী শোনাচ্ছে যে না, এই করোনা ভাইরাস থাকবে বেশ কিছুদিন, কতদিন থাকবে তাও বলা যাচ্ছে না এবং এটাকে সঙ্গে করেই আমাদের চলতে হবে, নানান রকম সাবধানবাণী শোনাচ্ছে। কখনো শুনছি যে করোনা ভাইরাস একটা জায়গাতেই স্টে করে থাকে। আমরা তো লেম্যান, এতটা বিষয়টা ভালো বুঝি না, আর আমি বাণিজ্যের ছাত্র। কখনো বলছে ভাইরাসটি একটা জায়গায় যতক্ষণ আমরা তাকে টাচ করছি বা তাকে ছুঁয়ে ফেলছি তাকে আমরা সেটা থেকে আক্রমণ হওয়ার সম্ভাবনা খুব কম। আবার কিছুদিন আগে শুনলাম যে এটা প্রায় 8 মিটার পর্যন্ত বায়ুর মাধ্যমে উঠতে পারে, অর্থাৎ বায়বীয়। কারণ আরো আতঙ্ক হচ্ছে, এই ধন্দ্ব আতঙ্ক এইগুলো নিয়েই আমরা খুব দিশাহারা। আমার মনে হচ্ছে এই ভাইরাসটি সম্পর্কে গবেষণার যে প্রথম দিক থেকে বা গোড়াতেই কোথাও একটা বোধহয় গলদ রয়ে গেছে, একটা ত্রুটি রয়েছে। প্রতিটি গবেষণার মূল ভিত্তি নির্ভর করে থাকে তার রোবাস্টনেস টেস্টের উপর, অর্থাৎ এটা একটা ফান্ডামেন্টাল, যেটার উপর ভিত্তি করে পরবর্তী পর্যায়ে পরীক্ষাগুলো চলতে থাকে পরীক্ষাগারে। তো এই রোবাস্টনেস টেস্টটা বোধহয় খুব ভালো করে হয়নি বা কোথাও গলদ রয়েছে। যারা গবেষক রয়েছেন তারা আরো আমার কাছে এটা ক্লিয়ার করে বলতে পারবে। আরো একটা সমস্যা রয়ে গেছে এই মিডিয়ার বদান্যতায়, যেটা আগে ছিল না। এত এত ইনফরমেশন প্রতিনিয়ত, প্রতি মুহূর্তে, এবং এর মধ্যে থেকে ফিল্টার করে কোনগুলো কারেক্ট, কোনগুলো ঠিক, এগুলো বোঝা যায় না। এগুলো আমাদের খুব সমস্যায় ফেলেছে। এর মধ্যে এটুকু তো আমরা ধন্দের কথা বললাম, কিভাবে চলতে হবে, ডুজ ডোন্টস নানান জায়গায় নানান রকম দিচ্ছে। কখনো লিস্টটা ছোট হয়ে যাচ্ছে, কখনো লিস্টটা বড় হচ্ছে, কোনটাকে ছেঁটে বাদ দেব, কোনটাকে রাখবো, বা সবটাই রেখে দিয়ে চলতে হবে কিনা, এইভাবে জীবন চালানো কি যায়? তো এতটুকুর মধ্যে একটা জিনিস, এই মাইক্রোবায়োলজি ডিপার্টমেন্টের সঙ্গে আজকে রয়েছি বলে, আলোচনায় অংশগ্রহণ করেছি একটু বলে আসার একটা বেশ ভালো সুখবর আজকে কাগজেই দেখলাম, যে মাইক্রোবায়োলজির পড়ুয়ারাও সামিল কোভিড লড়াইয়ে, এবং তারা বিনা পারিশ্রমিকে বেশ কিছু স্নাতকোত্তর স্তরের পড়ুয়ারা সপ্তাহে 6 দিন ঘন্টার পর ঘন্টা পিপিই পড়ি কোভিড পরীক্ষার নমুনা সংগ্রহ করছেন। এটা ওই মাইক্রোবায়োলজিক্যাল সোসাইটি অফ ইন্ডিয়ার ডাকেই তারা মাঠে নেমেছেন এই লড়াইটা করার জন্য। প্র্যাকটিক্যালি তাদের কাছে, অন্যান্য অনেকের কাছেই হ্যাটস অফ, এনাদেরও আমি সাধুবাদ জানাই। এখানে আইআইটি খড়গপুরের পড়ুয়ারা রয়েছে, লেডি ব্রাবন কলেজের পড়ুয়ারা রয়েছেন, সেন্ট জেভিয়ার্স ইউনিভার্সিটির পড়ুয়ারা রয়েছেন। তো আমাদের কলেজেরও অনেকে অনেক রকম লড়াই দিচ্ছেন। আমার জানাশোনা যারা রয়েছেন তাদেরও আমি ধন্যবাদ জানাই। তো এইটুকু বলে আমি আমার বক্তব্য শেষ করছি এবং তন্ময় ঘোষকে ফিরিয়ে দিচ্ছি মাইক্রোফোন টু কন্টিনিউ দ্য প্রোগ্রাম। থ্যাংক ইউ স্যার।
Information that Somo Sharathi Ganguly, he did his BSc degree from Dinabandhu Andrews College, and he is from IIT Kharagpur, and he is also member of Microbiology Society of India, and he doing this job very successfully, and we are all proud for him. And this is very privileged to say that he was our student, and he did his BSc from Dinabandhu Andrews College, Microbiology Department. Am I audible, sir?
Now it's audible.
Okay. Let me introduce to our speaker, Dr. AMARSHI Mukherjee. Dr. Mukherjee graduated from Surendranath College, University of Calcutta, with BSc degree in microbiology. He masters in Bidhannagar Government College, Calcutta University. He received PhD in biochemistry from NISET, University of Calcutta. Dr. Mukherjee, now a postdoctoral fellow at Johns Hopkins Medicine, Baltimore, in the Department of Rheumatology. As a Bengalis, we represent in foreign, proud to be a Bengalis as an Indian. We fully appreciate you finding the time to be with us despite your schedule. Now I am requesting you to share your valuable thought. Please, sir, continue your talk.
Okay. Can you hear me?
Yes.
Okay. Thank you very much for organizing the event. Thank you, Principal Sir. Thank you, Tanmay, and thank you, other speakers and the participants for make it valuable, make it to attend, and for organizing the event. So let me just tell you about brief of my research and what I'm doing here right now. So currently I'm working as a postdoctoral fellow in Johns Hopkins University School of Medicine. So I works on autoimmune disease, rheumatoid arthritis, but a group of researchers surrounding me, they are from different background, and Johns Hopkins Medicine is one of the hub of COVID research, and I think the world most accepted scenario is provided by the Johns Hopkins University School of Medicine. That means how many countries are affected and death rates, and we have a like group of really nice statistician who is collecting data every day and night for 24 hours and updating the whole world about the COVID-19 situation. So it's a privilege to work in Johns Hopkins Medicine, and I have a bunch of friends here who is directly related with COVID research as well as treatments, and there are like lots of doctors who are treating COVID patients. So when Tanmay requested me to present a webinar or seminar, so I contact a few of them, and before that I also shared their views. So I am a basics researcher. I work on basic science, mainly biochemistry, as well as some clinical research. So from this point of view, I will show you something about the COVID-19, and I'd like to share my screen. So let me show you that. Okay, so can you see my screen right now?
Yes.
Okay. So my today's topic is why is COVID-19 so different and dangerous? So I think the whole full screen view is available, right? So
Yes.
everyone can hear me right now. Okay. So my today's topic is why is COVID-19 so different and dangerous? I would like to introduce some basic thing about the virus in a very preliminary manner. Then I'll move to deep into the science to show what makes the COVID different than other virus and why it's dangerous. So if we look at the history of coronavirus, so people have idea that it's something new, but coronavirus is not new. So it's a group of virus or a type of virus that was first discovered back in 1930, and it was discovered in domestic chickens. Next, in 1960, people found that coronavirus can also infect humans. So there is a gap. So there's a gap of 30 years, almost 30 years or even more, 1930 to 60, and then it moved to domestic animals to human. And since 2003, at least 5 new human coronavirus have been identified, and some of them are really well known, like SARS and MERS, so which causes significant morbidity and mortality. Most of you know about virus and bacteria and cells and biology. So the audience is diverse, so I have decided to keep 1 or 2 slides to tell something very basic about the virus. Here in my slide, you can see that there is the reproduction of bacteria and reproduction of virus. So when bacteria reproduce, so bacteria are living entities. When they reproduce independently. So one bacteria generates into 2 different bacteria. But in case of virus, you can see here, this is the bacteriophage, the small one, which attacks a bacterial cell. So virus goes inside the cell. Bacterial virus affect bacteria, animal and plant virus affect animal and plant. They goes inside the host cell, and there they generate the virus particle, which assembles and finally lies the cell and comes outside. So the difference is that the virus are not living entities. They are dead when they are outside the host cell. They get activated or they become alive when they get inside the host cell. That is the really important thing we should have noticed, that viruses are not living. They are not considered as living when they are outside the host cell. Now, you have seen in this slide that this is the example of one animal virus that attacks sheep. There is the example of one plant virus that attack brinjals, and there are some viruses that attack bacteria like E. coli. So viruses cannot switch the host, like animal virus cannot goes to plant, or plant virus cannot goes to animals or bacteria. But the important thing is that animal virus or plant virus especially the animal virus, can switch one host to another host, not in plant or not in bacteria, but in animals. So this switching is called zoonosis. The zoonosis is any disease or infection that is naturally transmissible from vertebrate animals to human. So the important thing about zoonosis is that you can see here different uh, living beings like bats and migratory birds and travelers and cattle and different climate changes, and that carries the viruses from different source like cow and chicken and pigs, and from there it finally goes to humans. So when we start thinking about coronavirus, we should have keep this thing in mind that animal virus can transmit from one host to another. That is called the zoonosis. This is really important in case of coronavirus. Now, if we look at the history or the well-known data, we can see that mutations allowed zoonotic virus to grow in other hosts. Just let me tell you very brief about what is mutation and how the life goes. So our genetic material is called DNA or deoxyribonucleic acid, which codes for almost everything we have in our body. From DNA, it's a process called transcription and translation, which I find ultimately results in production of mRNA and other RNAs and proteins, and that finally build up our body. So our body is made up of mainly proteins and DNAs and lots of other chemical materials. And mutation is the change of DNA, which ultimately results in change of proteins, and different proteins has different functions. So you can see here that the origins of a different group of coronavirus, which are really well known, that is SARS, MERS, and SARS-CoV-2, means the novel coronavirus, all were originated from bats. This is already proved. Now, from bats, those viruses moves to different group of animals, and from those animals, it goes to human. So in case of SARS, it affects civets and raccoons and dogs, and then it's mutated and goes to human. For MERS, it mutated and goes to camels, and from camels, it goes to human. For SARS-CoV-2 or COVID-19, people are assuming that it's also originated from bats, and it's now proved that the main origin is bat, but the intermediate host is right now it's unknown. But finally, the virus get mutated and affecting the human. So virus going to get mutated, they have change of structure and they can attack different host. If we look at the statistics of other coronaviruses, so except COVID-19 or SARS-CoV-2, the most well-known are SARS and MERS. So SARS started in 2002 and MERS in 2012. If you look at the death percentage, in case of SARS, it only 9.5%, and with MERS, it's 35%, and coronavirus death rate is lower than SARS and MERS. But you can see in this slide significantly the affected people, number of affected people is really high, many times high, almost like 90, so 1 crore times affected people than others. So this is the main thing. That means the SARS-CoV-2 attacks human in a very high rate than other viruses. That make it more dangerous. If we compare, so SARS-CoV-2 or coronavirus 19 is a kind of virus that is similar to flu. If we take a comparative analysis of general flu virus that happens almost every year, and every year we got vaccinated in US for flu. If we compare these, we can find 3 different things. That means the basic reproductive number. If I just explain it in a very simple way, that means how many people the virus can attack. You can see here that COVID-19 has a high basic reproductive number, though the SARS also has a very high reproductive number, but COVID-19 is very high. The most significant thing is that the incubation time. So incubation time means how many days a virus or bacteria or any pathogen can stay inside the human body. You can see here that the COVID-19 can stay inside our body for 4 to 14 days. That means almost 2 weeks. That is significantly higher than any other virus. Community attack rate also represent that how many percentage of a population can be attacked by the viruses, and here you can see that 30 to 40% of the community is being attacked by the COVID-19. Those are the character of COVID-19 that is significant, and that's the thing that make the virus more dangerous than other viruses. Now, I would like to share some photos and videos with you. So, the privilege of working in Johns Hopkins Medicine is that I can see people from best people. I must say that best people from different part of the world, Asia, Africa, Europe, whatever. So, uh, in 2017, I joined in Johns Hopkins Medicine in 2017, and in that year, one guy came to our lab for 1 year training. He is a doctor, and he is from Wuhan, China. I'm not going to disclose his name because right now you know that we have some conflict with China, so as with China has some conflict with US. So Chinese people do not use Facebook, but this guy is my personal friend, so we used to chat through Facebook. But right now Facebook is banned in China, and Chinese people generally use another communication medium that is called WeChat, and currently we are not using WeChat. So I was not able to get a video from this guy, but this guy is a doctor from Wuhan, China. So all of you right now know that Wuhan, China, because people are predicting that's maybe the place of origin of coronavirus. But I know about Wuhan from 2017 because of this guy. So he sent me some messages that about the situation in Wuhan, China, and he said that I am just going to prompt that one. Hi AMARSHI, I have been working in hospital for over 2 months and cannot go home and rest in a hotel after work. I have not been tested COVID-19 nucleic acid and antibodies and chest CT image yet, only passed the test. Can doctors allowed to go back to home? I have not seen his son and mom for past 2 months, and we talk each other in Chinese Facebook, that is the WeChat, every night. That means doctors in Wuhan, they were not allowed to go home for 2 months, and they have like very tight working schedule, like 24 hours in 3 days continuously, then 1 day off, something like that. Initially, the pandemic suffering is badly catastrophic in Wuhan. God bless, eventually the spreading was contained and no more than 100 patients in ICU up to now. That means they are able to manage the disaster, and they did it in a really, really strict way. But most are in critical conditions. Some are in mechanical ventilation, and some are being supported by supporting system. The chances of survival is small. I think we have persist 2 or more weeks. From this news I got that the mortality in New York is high, and whatever he mentioned that masks are so important. I heard that Americans don't like to wear masks. That is somehow true. Actually, that is the most effective equipment to combat the virus. So people have questioned, such in the beginning, Dr. Professor Tanmay said that how to combat with the virus. So you got an opinion from expert who is from Wuhan and who is a doctor there that the best thing is the mask. So wearing a mask is most important. So in my hospital, no staff or faculty in the respiratory infectious disease department got infected with the adequate PPE. So if you have the preventive measure, you must be safe. Well, there were 6 surgeons confirmed case because they didn't wear mask. So point to be noted that you need to wear a mask. So I'm going to show you some images. So I didn't get permission from that guy, but I think I didn't disclose his name or his photos. So this is the images from this is rare images because Chinese are not allowed to send their photos outside their country. So this is the images of hospital in Wuhan, COVID treatment group, Wuhan, China. So I had one friend who came to our lab for 2 months. He is a doctor from Milan, Italy. So I have decided to include one video of him because he directly deals with COVID patients, and I requested him for a video and he sent me the video. So I think you will be happy to hear the video of Dr. Stefano Rodolfi. He is a doctor from Milan, Italy, and he treated COVID patients. So let's go to see what Stefano says.
Hello everyone, I'm Dr. Stefano Rodolfi. I'm a resident physician in internal medicine in a big hospital in the area of Milan. I'm making this video to share my personal experience as well as my opinion on the COVID-19 pandemics. As you know, Italy was the first European country to experience a big COVID-19 outbreak at the end of February, beginning of March. And the most affected area was the northern part of Italy, Lombardy region especially, so Milan as well. So when it all happened, the first thing that the first striking problem in coping with the COVID-19 pandemics was lack of proper structures. We were literally overwhelmed by COVID-19 cases. Hundreds of patients accessing the emergency department for respiratory insufficiency due to viral pneumonia, and completely saturating the emergency department, the hospital wards, the intensive care units of a lot of hospitals. So the first thing that made incredibly difficult to cope with the pandemics was lack of structures. The colleagues that were working in the emergency department had to witness scenes of patients dying of respiratory insufficiency without even being able to be visited by a doctor, because of lack of personnel, lack of sufficient personnel. Some patients that had to be put on sedon analgesia because they couldn't access a mechanical ventilator, even not even a non-invasive mechanical ventilator, because they were already been allocated to other patients. Moreover, in the first periods, we had the mortuary room that was completely full in a lot of hospitals. So corpses of patients who died for respiratory insufficiency had to stay in the emergency department for all night long, for example, nearby live patients. So these are stories that really that are really difficult to hear. That were really difficult to be told as well by my colleagues. As to my personal experience, I worked in a hospital ward of COVID-19 patients. It was really difficult to take care of these patients, first of all, because in the beginning you felt you weren't able to do anything. Everything you did couldn't change the natural course of the disease, and as we can see, we didn't make much step further even now. So we had to try and cure the best way as we could a lot of patients that then progressed to death due to respiratory insufficiency. As to my personal opinion, I am in favor of the lockdown maneuvers in Italy. I think they worked perfectly. Maybe we were late in performing lockdown. We didn't track the first cases. That's one limit for sure. But being able to lock down the entire country for 1 month, 1.5 months, gave hospitals the chance to slow down a little bit, to be able to have enough resources for all the patients. And as we can see now it's been almost 1 month since the reopening, and we are witnessing a lot less cases. We can say to be out from the first outbreak. I am I am I'm sad. I'm really sad to be hearing bad news from the colleagues working in the United States, as well as bad reporting news. I think that in the United States as well, maybe now it's also too late, but I think that being able to perform strict lockdown measures could really help slow down the contagion curve, which in our experience was the striking part to be addressed. I end this video with a big hug and a big wish for all the colleagues working in the United States. Hope that you can get out of the COVID-19 outbreak as we did. Goodbye.
Okay, so we have just shared the opinion of an Italian doctor, Stefano. So he supports lockdown, and as well as so you have to wear a mask all the time. So I think his opinion is really valuable, and you can see that while lockdown is important, but long-term lockdown is not good for people who are like poor or below poverty level. So you have to go outside, but always wearing a mask. So let's go deep into the science, and this is the structure of coronavirus. And you can see that the virus is made up of some membranes and some nucleic acid inside the membrane. So virus has only one type of nucleic acid, either DNA or RNA. So it has different proteins, and I have marked one in red that is called spike glycoprotein. So the blue one in the image. So this one is important because this is the glycoprotein which binds with the cell surface receptor of humans. That means that that's protein help the attachment of the virus with the human host. So I will go more detail about that. And here I will give you a brief life cycle about the virus. So the virus binds with a human cell, and for binding they need a specific receptor in the human cell. The receptor is called AC2 receptor. So the virus attach with the AC2 receptor, then it get internalized into the cell. It uncoats its covering and release the nucleic acid, which is RNA. Then it generates multiple copies of RNAs and finally proteins, and form the virus particle and travel throughout the cell. When the virus particles are assembled, they lyse the cell and go outside. So I'm not going to describe it in detail, but the main thing is that the virus bind with some cell surface receptor. The glycoprotein bind with the AC2 receptor. That's why virus goes inside the cell, and then there inside the cell, the virus replicate, form other virus particles, and burst the cell or lyses the cell and comes outside. Now, modern days science are interdisciplinary. That means peoples of different field are working on different projects and same branch of the science. So one field is called structural biology, which shows how the structure of macromolecules looks like. Microbiologists, biochemists, structural biologists, doctors, and immunologists all are working together to explore the fundamental biological problem. It's maybe a virus or bacteria. So we have one friend here in Johns Hopkins who is a structural biologist, and he is actually from Germany. I asked him to share his idea about the COVID-19. So he gives some valuable feedback about the condition in Germany and how they are able to manage the COVID-19, and give some very nice idea about the virus surface glycoprotein. So let's switch to Nick video.
Yeah, hi there. My name is Nicholas Hoffman. I am a postdoctoral researcher here at Johns Hopkins University in Baltimore, Maryland. And I'm very glad to have the opportunity to say a couple of words to you regarding the pandemic and the severity of this virus. A little bit of background. I come originally from Germany, and I did my German diploma, which is basically a master's in Tübingen in biochemistry. Then I transitioned to the European Molecular Biology Laboratory in Heidelberg, where I did my PhD in structural biology. I focused during my PhD on mostly electron cryomicroscopy, and this is what I do right now here in Johns Hopkins. I work on gene expression of transcription factors that associate to nucleosomes using cryo-electron microscopy and a variety of biophysical methods. With regards to the virus, obviously the situation is very severe. As you might have heard, some countries have handled the outbreaks that have basically happened all over the world better, and other countries have done much worse. Here in the US, the results are very mixed, but overall could be better. In Baltimore and the state Maryland, infections are currently going down, and this is basically the second week that we are allowed to go back to work at reduced capacity, wearing a face mask, which I took off for this video because I'm in my own in the room, but as soon as somebody comes in, of course, I will put it back on. In other parts of the country, it's looking very bad. In the south, infections are currently rising, and we are getting back to a serious outbreak here with unknown consequences for health and economy for everybody. And the situation is similar in many parts of the world. In my home country, Germany, the situation looks better. The general outbreak is under control, and instead there are small parts and pockets where local outbreaks still happen, and the country is trying to handle this with regional lockdowns. Now, I'm not a doctor, but I think following the science behind the outbreak, there's a couple of things that really help to prevent strong outbreaks and to keep you safe. The most important thing is to wear a mask. If everyone wears a mask, which is, for instance, the case in Asian countries like Japan and China, the outbreak is almost completely under control. And the reason is that even a cloth mask prevents people that have the virus to spread particles through the air that will infect others. And if nobody spreads particles, people have a much lower chance of becoming infected. Number two, social distancing. Don't spend too much time with your friends in a closed room at too close distance. Instead, try to meet outside in the park or in the backyard, and only take off the mask when you are far apart. And number three, of course, wash your hands. Now a little bit on the science before this video is over. My profession, structural biology, can contribute greatly to better understanding how the virus works. And if you're interested, I want to bring your attention to one publication from Seattle, which I have prepared here, from the Wiesler lab. And what they have been doing is they were able to solve structures of the virus spike protein, which is absolutely essential to understand how the virus enters the cell and is the future to providing a vaccine against the virus. With this, I would like to finish this video. Stay safe, and all the best to all of you. Take care. Bye-bye.
Okay, so a really nice and thoughtful videos. So he gave a brief about the condition in US and Germany, and he also mentioned that wearing a mask is really important. Germany is able to handle the situation because they are very strict on wearing a mask, and they have also imposed lockdown, but things are going back to normal state in Germany, who are really strict on rules and regulations. So here I am going to show you something that is more scientific and more deep into the science, that how we can see a virus and how it helps to build up the vaccines. And I think tomorrow Srijani is going to tell more about the vaccine, but I'm going to share something that is why this coronavirus is dangerous and what is the role of the structure in that process. So that is more scientific. So if you look at the image at the left, that one artist is painting something on her canvas. So it's the image of a nature, and she's just drawing that image on her canvas. So the canvas is the model and the real thing is the environment. Now, if you just look at the image of the right panel, you can see a small round here. That is the actual image of the virus that you can compare with the real nature image. Then they have enlarged the image, and next they build up the models. So cryo-electron microscopy is an important tool to see the structure of viruses as well as any biological macromolecules. So the image we can see is the models that is built up on the real image, just like you can imagine a painting on the canvas that mimic the real nature. So here from the enlarged image, they build up the model of the some surface protein, and here you can see the models that contains different parts of the proteins. So if we look at the fact, what scientists have discovered so far, and has no controversy about that, because lots of people are working with COVID now, and they are every day they are proposing something new. But these are the thing that is really well known and well established, that the virus has a primary surface glycoprotein, which Nicholas already mentioned in his speech, that binds with the receptor that I have mentioned earlier on the human cell, that is called the ACE2 receptor. So virus bind to the receptors of human cell and then it goes inside. The second one, the affinity of the virus glycoprotein with that receptor is really high, and higher than any other coronavirus species. So that means the virus very tightly bound with the human cell. That's why it's spreading in so high rate. The third one, so if we are infected with some virus or bacteria, our immune system generates one molecule that fights with infection, that is called antibodies. So when the SARS-CoV virus affect the human, the previously existed antibody that attacks other versions of COVID doesn't recognize the new COVID virus. That means our immune system doesn't recognize the COVID-19 virus. So we are not very well equipped to fight with that virus. So these are the very well explained and well established reason about the COVID-19. So I'm going to tell you in more detail about that. So this is the cryo-EM structure of ACE2 receptor binding region of spike glycoprotein. You may recap that the virus has some glycoprotein on its surface. So at the left, you can see that surface glycoprotein of SARS-CoV-2, and this is the SARS-CoV-1, that is preliminarily known as the SARS. So if we overlap the green region with the white one, you can see that these two are not identical. That means this virus glycoprotein has mutated, and the mutated version is different than the previous version or older version. So the green one is quite different than the previously known virus structure. Now, if we just look more closer view of the whole thing, that means the top part of this region, and build up a model. So you can see there, you don't have to be very scientific to see that one. You see that one white color thing and one red color thing. So the white color thing represents the SARS virus receptor binding region, and the red color thing it denotes the COVID-19 receptor binding region. So you can see that there are some significant difference in this highlighted area here that's marked by red. That there are lots of red patches on the top of the region. That means these patches are new. That means these binding regions are new in COVID-19, and this region contributed severely with the binding with the ACE2 receptor. That means people says that this virus is very sticky. Why this is very sticky, or why it binds with the human cells because it has some modification on its surface glycoprotein that makes it more potential to bind with the human ACE2 receptor. A group of Chinese people, I'm going to explain it in an easier way, that they have did some analysis of different viruses and their amino acid sequences. So amino acids are building block of proteins, and those amino acids are responsible for binding with the surface of the human ACE2 receptor. So here you can see that they have analyzed a bunch of viruses, SARS-human, SARS-CoV, and they have found the red colored things, that means the mutations. And you can remember that in one previous slide, I have mentioned about the mutations and how mutations help the virus to switch the host from dog or bat to human. So they have mutations in some amino acid. This 1 year latest represent the amino acids. And you can see that the red colors are the mutation. That means virus has some mutations in amino acid, and these mutations make them more potentially attached to the human ACE2 receptor. The green colored thing is an image of human ACE2 receptor, not image, but the model of human ACE2 receptor. And the violet one is the virus. And you see that the amino acid looks same in human ACE2 receptor, which is conserved. And here you can see that the binding region of virus has changed. And the left one, this one is the SARS-CoV-1, and this one is the SARS-CoV-2. If you see that, you can see the amino acids are different. That means that in the binding region, amino acids mutated. That's why it binds more tightly with the human cell receptors. That's why it's spreading so much, because the surface glycoproteins has been changed. Now, the most important thing of immunity is the antibody response. So there are a bunch of people who had COVID-1, or that's called SARS, and they are affected by SARS and survived. So a group of scientists tested those people who was affected by the SARS and collected their antibody and checked the antibody response against COVID-2 or coronavirus 2. But they have find a surprising result that the previously immune people against SARS-1 cannot recognize the SARS-2 virus or the coronavirus. You can see one straight line here. That is the antibody response of against coronavirus 2, and the straight line means there is no change in antibody response. So if the line goes up high, that means the antibody is working. But in 3 different antibodies, you cannot see any change of the straight line here. That means the antibody that is already generated against SARS coronavirus cannot recognize the SARS-CoV-2 or coronavirus 2019. That is really important, because virus has modified itself by such a way that previous antibody response cannot recognize the virus. That's why people are not immune against that virus. Now, so we have one friend who is an immunologist and works with immunity and this kind of thing, who is actually from Spain. So I would like to share her opinion with you. Let's see what Martha says.
Hello, my name is Martha. I'm an immunology PhD currently working as research specialist at Johns Hopkins University. The novel coronavirus, COVID-19, has already spread around the world at a rate that nobody would have imagined. Some people still believe, is it a virus like flu? Even both are infectious respiratory illnesses, and despite the symptoms can look similar, virus COVID-19 is, as far as we know, more dangerous and deadly than flu. COVID-19 not only infects and damages the lungs, its reach can extend to many organs, including heart and blood vessels, kidneys, gut, and brain. It can attack almost anything in the body, with devastating consequences. If we take this in a big picture, we are not talking about only how many people can die, also how many people can end in the intensive care unit for weeks, how many people can be chronically infected, and also can affect at all those people with other diseases that cannot be treated as we used to do, like doing regular checkups, surgeries on time. So each of us have a huge responsibility to prevent the spread of the COVID-19. How? Covering our mouth and nose and with a face mask when around others, cleaning our hands often and keep distance between us. Be responsible. All are involved in this.
Okay, so she said about something that means the virus can attack different part of the body. So it is really important to know one term that is called tissue tropism. That means a cells and tissue of a host that support growth of a particular virus or bacteria. We know that there are different parts of our body which is affected by different virus or bacteria. The advantage of COVID-19 is that it can attack different part of the body, because the ACE2 receptor is found in brain, lung, kidney, intestine, and heart, and different parts of the body. So the virus can spread. It's not restricted only in the lung, but it also affects brains. The report has been found, and it can affect kidney and lungs and other parts of the body. So this is one really means think really matter that we think we should think about that. That's its affect different part. Another thing is that the symptom, if we have flu or some other kind of viral infection, that's restricted in fever, cough, and cold, that kind of stuff. But here you can see that lots of symptoms, and that's why people are very frequently talking about asymptomatic patients, because it has lots of symptoms. I have met some people here in Hopkins that who didn't have at all any symptom of COVID, but they were tested positive, and some of them only had like they don't have any taste. That's the only symptom they had. So it's a really annoying and really means bad thing that the virus sometimes is turning out to be asymptomatic. Now, so people have some questions about for how many hours virus can stay outside. Lots of people are working on that, but the paper that was published in New England Journal of Medicine, which is one of the best journal of the medical field, they say that it's not something really different than SARS-CoV-1. If you see the comparison here, that SARS-CoV-1, that's the SARS virus, and SARS-CoV-2 means the novel coronavirus, you can see that both stay in the aerosol for 3 hours, both stay in the plastic for 72 hours, and stainless steel 48 hours, and no attachment on copper or cardboard. So it's not very significant that a virus stay outside longer than other viruses. So SARS-CoV-1 has the similar symptoms. Then what make it so different? So they have predicted that, indicated that the main thing was that virus probably arise from other factors, including high viral load. That means the virus replicate very fast and stay inside the body. So when it goes outside, the amount of virus increases in a very high number. That's called high viral load. That's maybe the one main reason why the virus is so dangerous. Now, a group of researchers at Johns Hopkins means they have discovered that there is one very severe effect that called cytokine storm. So cytokines are the molecules our immune systems secrete those cytokines, then cytokines molecules fight against the infection. So I think tomorrow Srijani is going to discuss about that in detail, but what I'm going to tell you that in brief. So if the cytokine response is not very well controlled, it may cause death. That's why Professor Andrea Cox, she has mentioned that it's like trying to kill a bug with a hammer. You can put a hole in the wall while you are trying to smash the bug. That means if you are trying to kill something, it may damage your own thing. So that's why sometimes the cytokine response is very high, and it's affect the people. That's huge cytokine response killed as well as the virus and as well as the people. So it's mostly found in the older people. Now, recently, WHO is being very controversial, and they have mentioned that acknowledge that there is an emerging evidence that coronavirus can be spread by tiny particles and suspended in the air. So that is not confirmed. Who share their opinions, and that is based on several research. So researcher says that it may stay in the air, but the thing is that if you are in open air, the virus cannot stay for a long time. I think day before yesterday, I got one email update from Johns Hopkins University. They did one analysis, and they have mentioned that in jail, in prison, the virus is 5.5 times more active because the air is not circulating. So the thing is true for confined air. That means where the air is not circulating, maybe the virus can stay there in the stagnant air. But in open area, I think it's not likely that it could be spread by the air. The second one is that who claimed that asymptomatic patients are not spreading the virus, and it's really contradictory, because after that they have mentioned that we don't actually have the answer yet. That is true, because asymptomatic patient, that means who don't have any symptom, they don't even know that they are COVID positive, they are the really means huge risk for the society, because they are also spreading the virus. That's why social distancing and wearing a mask is so important. If everyone wear a mask, so I think that will minimize the problem. The next one I'll show just a part of the history. That means this kind of pandemic happens before, and so the 1981 Spanish flu that affected almost 50 million people, and lots of people died. So compared to that, coronavirus is not so severe. And there are other pandemics in 1950s and 1960s and 2009. So those pandemics were also, when people recovered from those pandemics, and it took 2 to 3 years for the recovery. And I hope that coronavirus pandemic is going to be end within few years. But we need to take precautions like wearing the mask. So this history shows us that we should have been positive and wait, and after a time everyone will be immune with that virus. So in a conclusion, so I can mention that why the virus is so dangerous is that change of surface glycoprotein structure, which make the virus more sticky, and that's why it binds with the human cell surface more tightly, and that's why so many people get infected. Long incubation period, it can stay inside the body for up to 2 weeks. That is really high for a virus to stay inside a body. Broad tissue tropism, that means the virus can affect from different tissues of the body. It can travel from one tissue to another. Variation in symptoms, the asymptomatic patients, and a very high viral load. So those are the specific things. That's why the virus became so dangerous. And I will answer any question, and I just really like to thank my friends and my family, especially the good guy from Wuhan and China, Nick, Stefano, and Martha for their valuable input. And I would like to thank my BSc teacher who taught me that science is beautiful. And I'll be really happy if you ask me questions, and let's go to the question answer session. Thank you.
Thank you very much, Dr. Amarshi Mukherjee. Now, question answer session is begin. Please ask some question if you have any inquiry. How many time COVID-19 long-lasting in disease?
Well, so they have tested some cottons and that kind of stuff, but it shouldn't be more than 72 hours. The maximum time period is 72 hours. And that's why we wear aprons, and aprons need to be cleaned in a regular manner, like within twice in a week. So it shouldn't be more than 72 hours. And as you know that viruses are non-living, they generally don't survive outside environment. And The paper I have mentioned, the New England Journal of Medicine, they have shown that a virus can live outside the host cell for 72 hours, but the number decreases with time. So first 24 hours, almost 50% virus particle dies. Then with time, it decreases. So it's not more than 72 hours.
Okay, thank you. And the second question is, how long the virus become active in asymptomatic?
Very good, very asymptomatic patient. So very good questions because they gave a range. That means in asymptomatic patients, it's maybe 4 days, 5 days, and even 10 to 12 days also, because they don't know, and who knows that who is asymptomatic. So people just randomly test and they found that they are positive, but they don't know when they get positive. So the range is not very specific. It's 4 days or maybe 10 or 12 days, but they don't have any symptom. That's why they really don't know how many days they have been affected. Okay, next question.
Next question is, sanitizer or soap, which is better precaution against COVID-19?
I'm not very clear about that. I think it should be soap. Okay. So sanitizer and soap. I must suggest soap for cleaning your hand, because sanitizer are also good, and all of them demand that 99.9% of bacteria and virus get killed because of the sanitizer. But soap are always better because you are cleaning off everything from your hand. So sanitizer will kill if there are some virus or bacteria, but soap will remove everything from your hand. So I think soap is better than sanitizer. Sanitizer are always good, but soap is better. Is there any proof of asymptomatic patient can affect after 14 days of isolation? Well, so this is a really nice question, and there are some controversy related with that, because some patient had been tested positive after 14 days, but those incidents are really few. So if we make a general conclusion, that doesn't show any sign after 14 days, but there are some exception which shows that they are COVID positive after 14 days. So that's the answer. Okay. So what are the difference between influenza virus and COVID? Well, so viruses are different. So influenza virus is a type of virus which cause cough and cold, and COVID is a different class of virus that also cause cough and cold and other fever, that kind of stuff. So each viruses are different. So basically, if you just look scientifically on it, so it's the structure, it's the genetic material, all are different. So one virus differed by another, but there are lots of ways to differentiate between them. But they have some common symptoms. That's only the similarity. Otherwise, they are from different group of virus. Okay.
Sir, will a single vaccine be enough for COVID treatment?
Well, so we don't have any vaccine yet, and who knows that whether we need a single vaccine or multiple vaccine. I think Srijani, she is going to give you more idea about vaccine, but I would like to mention one thing, that vaccine are updated almost every year. So in US, we need to vaccinate it every year, and before winter, and it's mandatory. If we don't take vaccine, they will stop our salary. So every year the vaccine, they change the vaccine, they modify the vaccine. So I'm not sure that whether one vaccine is sufficient or they need multiple vaccines. So we have to wait for the answer. Okay. Can you explain how does the spike glycoprotein, sorry, could you please repeat the question, please? Changes its shape in response to antibody. Well, so spike glycoprotein doesn't change its shape in response to antibody, rather it's already changed. So it's already mutated. That's why it has a different conformation that is not recognized by previously existed antibody. So virus do not change its antibody structure when it's surface glycoprotein structure when get encountered by the antibody. But it's the version that we have right now already changed their surface glycoprotein structure. That's why antibody cannot recognize that, and I hope that in future we will get some antibody response against the virus. Okay. How virus is removed from grain vegetable? So everything you can remove by cleaning or by washing, and the best way is to wash by water, because water is the best solvent. And some people are dipping their vegetables in soap water, but I think it's not absolutely necessary. And the people, what they used to do here, they just buy fresh vegetables and keep them outside for 2 to 3 days. Then that's why they think that the virus will die, and then one cleaning will kill all the viruses or remove all the viruses. Next question.
I think.
Okay.
Oh,
Does the temperature affect the coronavirus? So, well, you know that it started in February, and that is cold, end of January and February and December. So it survives in cold temperature, and right now it's middle of July. So the virus, so when it stays outside the host cell, I think it's not being affected by the temperature, because inside the host cell or inside our body, the optimum temperature is 37 degrees centigrade, and virus can tolerate. And but I must say one thing, that in cold temperature, it's maybe slow down, but it's not severely affected by the cold temperature. It's already proved.
spike, H and
Okay.
hemoglobin are same type or no?
No. So I have already show that there are different proteins. So these proteins all are different. So different 5, 6, or 10, or maybe different types of proteins are present in the virus. All are different, but their location. Some spike proteins are surface glycoprotein, and they have some protein that is inside the virus particles. So those are different. Okay.
Really good.
Definitely, it's a really good question. That can animal carry the virus that causes? Yes, that's the one reason. That's why the virus is spreading. So that's why it's possible that the virus can stay on the surface, on the fur. That's possible. That's highly possible. Mhm. Any more questions?
I think
Okay, pretty much done with the question and answer. So,
Romi madam is there. Hello, Pronomi madam is there. Hello.
Yes.
So first and foremost, I would like to thank the Department of Microbiology and all the faculty member of Microbiology and IQAC for arranging the most wonderful webinar. I thank from core of my heart to Rishin Bhattacharya, mother-come student, for supporting me and us for cooperate with this webinar. Once again, thank you all. And very, very thanks to Dr. Mukherjee for give us your valuable time. And if any faculty tell, you can tell something.
okay.
So,
Okay.